Thanks, Rachel. Thanks, Rachel, and thanks for the invitation to give the last webinar of the term. So when I was thinking about what I wanted to share with you today, I decided to cover a recent paper we had come out in Nature, Ecology, and Evolution last year, looking at the biogeographic context of human evolution in Eastern Africa over the last 6 million years. And so what I'll do today is walk through the motivation behind this study, how we went about it, and why this new biogeographic context allows us to ask questions about the relationship between environment and human evolution in a way we haven't really been able to do so before. So I want to start here. As many of you know, 2025 is the centennial of the announcement of the discovery of the Taung child in South Africa and the naming of the first truly ancient African hominin taxon, Australopithecus africanus. This is what hominin diversity looked like in 1925. You basically had A. africanus giving rise to a few species of Homo. And in contrast, this is where we are today in 2025. We have quite a rich record of earlier hominin taxa that we didn't know about in 1925. So various species of Australopithecus and this group in green, even earlier candidates for being early hominin taxa. And what is really interesting for me about this picture as it's developed in 2025 is actually how crowded various periods of human evolution are with multiple species coexisting at the same time and in some cases in the same place. And so what I want to highlight here is the record between 3 to 4 million years ago, where we have in Eastern Africa, maybe 3, 4, 5, Eastern and Central Africa, I should say, 3, 4, or 5 different species in coexistence. So the areas I'm talking about are the Chad Basin and Central Africa, and then in the East African Rift System, the Afar of Ethiopia, the Turkana Basin of Ethiopia and Kenya, and the Southern Rift area of near Laetoli, Olduvai, Olorgesailie, etc. If we look at these areas between 3.5 to 3 million years ago, we begin to see an interesting pattern in terms of the distribution of these putative hominin taxa. I say putative because some are confidently established, like Australopithecus afarensis, others not so much. And so if we take the record at face value, we have Australopithecus afarensis known from both the Afar and the Southern Rift region. In the Turkana Basin on the west side of Lake Turkana, there is what is argued to be a distinct taxon, Kenyanthropus platyops. In Chad, again, something argued to be distinct from A. afarensis, Australopithecus barrel-ghazali. And even within the Afar, at the site of Woronzamile, at the same time, there have been recent arguments that the jaws and teeth from this 3.5 million year period represent a distinct taxon, Australopithecus deiremida. And in addition, there's this really interesting foot that has an opposable big toe, the Bertelli foot. And so there's some evidence for hominin diversity at this time. And although it's controversial, it's in some cases doubtful that some of these name taxa may be distinct from A. afarensis, but that's not the focus of the talk. What's interesting to me is that if we take that record at face value, there's a clear biogeographic structure where the taxa we're finding in the Afar are different from Turkana and from Turkana, from Chad, etc. And so that got me and my colleagues thinking, what might what role might biogeography have played in shaping the diversity and distributions of hominins, both in that mid-Pliocene window between 3.5 to 3 million years ago, but also later in time. And so when we're thinking about biogeography, we're thinking about the factors that govern the diversity and distribution of life on Earth. One of the founders of biogeography was Alexander von Humboldt, who noticed that physical geographic characters like climate or topography played a really important role in shaping diversity along mountain ranges, for example, in South America. And then most of you will know Alfred Russel Wallace as Darwin's co-discoverer of natural selection. He also was one of the first true biogeographers. And so this is Wallace's map of the major zoogeographic regions of the world, where each one of these colored areas is host to a very distinct assemblage of animals. You can do this with plants as well. So here's an updated map of Wallace's zoogeographic regions from 2013. And you can see that in Africa, North Africa forms its own biogeographic province, the Sahara-Arabian province with Arabia, whereas sub-Saharan Africa is all Afrotropical. We can go a step further and partition out subregions within Eastern Africa. So this is a study by Peter Linder and colleagues in 2012, where they looked at the distribution of both plants and animals across sub-Saharan Africa, and using clustering algorithms defined biogeographic subregions. And so those are the colored polygons you're looking at on the map here. I think the most interesting thing about biogeographic patterns is that they tell us something about history, about evolutionary history. Biogeography, the outcome that is biogeography today, really represents the interface between ecological processes, evolutionary processes, and time. And so that's not surprising. Most of us know this from the time we're undergrads. When we learn about factors, evolutionary factors or processes like dispersal, speciation, extinction, most of those processes have a geographically explicit aspect to them. And so, for example, with allopatric speciation, we have a starting population or species, in this case, A. On the left, we're looking at it through time. On the right, we're looking at it through time and space. If we get a physical geographic barrier that limits or weakens gene flow, the constituent populations on either side of that barrier will begin to diverge. If they stay isolated, we get local selection, adaptation. We also get genetic drift. And ultimately, by the end of this process, we end up with two distinct species that have arisen through basically geographic separation and the separation of the lineages through time. We can see these processes playing out, or evidence of these processes playing out in biogeographic patterns today. So this is from a really nice study by Eleni Lorenzen in 2012, where she and her collaborators synthesized all that we had in terms of phylogeographic data. These are genetic data on population structure for large-bodied savanna-dwelling African herbivores. So what the map to the right shows, or the series of maps, are different large-bodied herbivore species alive in Africa today and how their population genetics are structured. And so you can see in many of the groups or many of the species, there's this really clear east-south divide where the populations in blue in East Africa are genetically distinct from those in Southern Africa. And they linked this patterning that we see across a huge range of taxa to past environmental change. We know that in the later Pleistocene in Africa, when things become very arid and dry during glacial phases, savanna vegetation expands at to the expense of forest, whereas when things get very wet and humid, like the African humid period, the forest belt that today is restricted in central and West Africa actually extends further east into Eastern Africa, as you can see on the map to the left. And so this push and pull of the savanna and forest biomes across the belly of Africa is what Lorenzen and colleagues argued was the driver of this particular population structuring. And so, again, just by looking at population structuring in relation to geography, we're getting some idea of the underlying role of environment in shaping these patterns. Biogeography also allows us to look at speciation in action. We can't observe in a human lifetime speciation happening, but using geography and patterns of relation, we can infer it happening. And so this is a map showing the distribution of subspecies of hartebeest. Each one of the colored polygons is the range of the species. And if you do genetic studies, this is from mitochondrial DNA on the right, they actually cluster into two predominant lineages. There's a northern lineage and a southern lineage that split about 1 million years ago. And then in the northern lineage, there's a further division between those in the east and those in the west. And so what we have basically is the pattern of relatedness among these hartebeest taxa mapping onto their geography. It's showing us speciation in action. When we have these complexes of species, or in this case, incipient species, they reveal to us the role that geographic differentiation plays in evolution. The last example I'll highlight here is the distribution of sister species. So if we go to taxa that we are pretty confident are distinct species, we again see this really interesting geographic pattern where one of the sisters is more northern, has a more northern or eastern distribution. The other has a more southern distribution. And again, we can probably link this to that recurrent expansion and contraction of forested versus savanna environments during the Pleistocene. That's basically the engine of speciation for a lot of these large savanna-dwelling African mammals. At least that's what we think. As well as areas where we have really high diversity. So many of you probably have heard of the refugia models for Pleistocene refugia. This is a map from Jonathan Kingdon's book, Island Africa, showing hypothesized areas where there's a desert refuge and a forest refuge that would have hosted the last remaining pieces of their respective environments during either really arid or really humid phases of the Pleistocene. So we think we have a pretty good idea of the role of recent, and by recent I mean the last few 100,000 years, of climate change in shaping biogeographic patterns of species diversity and distributions through time. But we actually know very little about long-term biogeographic patterns in Eastern Africa. By long-term, I mean those that played out over millions of years as hominins were evolving in the East African Rift system. And if those patterns are changing through time, those biogeographic dynamics, what were their underlying environmental drivers? And so that's the question we really became interested in this study, because having this interesting pattern of putative early hominins in different places at the same time, that's really interesting. But it would be informative to know what the other large mammals were doing at the same time. Would we expect this high degree of endemism where the taxa in the Afar are different from those in Turkana, etc.? So that context becomes really important. It's not direct evidence as to whether these things on the screen may or may not be the same species, but it feeds into that debate. And so that was basically the overall goal of our study, was to provide for the first time a really long-term, statistically defined biogeographic context for Eastern Africa. So what we did was we went out to the literature and museums and collected data on paleontological assemblages from 3 areas of the East African Rift system. Each one of these white points on the map is a fossil community or assemblage. So, for example, the Tulabor member of the Kubifora Formation in East Turkana, the Denindora member of the Hadar Formation in the Afar, etc. We ended up with about 100 fossil communities total across these 3 regions, spanning the last 6 million years. And we focused solely on the terrestrial large mammals. So these are things like antelope and horses and elephants and carnivores and monkeys and hominins, anything that weighed over a kilo and was a terrestrial mammal that was in the fossil record we included in this study. The way we quantified biogeographic change through time was through spatial beta diversity. And beta diversity is basically a way of quantifying in a standardized way compositional differences among different communities. So here we have three communities, 1, 2, and 3. And as you can see from the shapes inside, they all contain the same 5 species, the blue triangle, the red triangle, etc. We can, using this example of minimum community differentiation, calculate some variables. So alpha diversity is average species richness at the community level. So in this case, our alpha diversity is 5. Our regional diversity, how many species exist across the whole blue box, is also 5 because each community has the same species. And therefore, our value for beta diversity, which is just the ratio between regional and local diversity, is 1. We may have 3 communities spatially, 1, 2, and 3, but because they're completely redundant, beta diversity is 1. They're all identical. We can then consider the other extreme where each community is, or hosts rather, a completely different set of species. And so this is our maximum differentiation example, where our alpha diversity is 5. Each one of these communities, 1, 2, and 3, has 5 species, but each one is distinct. And so our regional diversity is then 15, and beta, being the ratio of regional to local diversity, is 3. So it just gives us an idea of community differences or compositional differences among the communities for a given region. So the particular beta diversity metric we chose was the multi-site similarity index defined by Baselga and et al. In 2007. And so you'll see that referred to here as BSIM or beta sim throughout. This metric is bound from 0 to 1. 1 means that you have maximum differentiation. Each community is composed of a different set of species. 0 meaning they're all redundant and they all overlap. We chose this measure for a few reasons. 1, it allows you to simultaneously compare beta diversity across the 3 regions. Some of the indices can only do pairwise comparisons, but really the big advantage of going with the multi-site Simpson beta diversity index, the Simpson index, was that it accounts for both the nestedness and the turnover components of beta diversity. And so I want to point you to the figure to the right. In the top panel, we have site A, B, and C, and the differences in the species that they share are the result of turnover, changes in species composition across space. So site A has 4, 5, and 6, B has 7, 8, and 9, C has 10, 11, 12. That's a quite different pattern than what we see below, where site D has 1 through 6, site E has 1 and 2, and site F has 1. That's nestedness, where 2 of the sites, F and E, are simply just subsets of the more rich one, D. We really liked the ability to partition out turnover versus nestedness because when we compare fossil communities that differ in degrees of preservation, how well they're sampled, it's very likely, or it can be your null hypothesis, that differences between those communities simply reflect ones or the smaller ones being subsets drawn from the larger one. And so the ability to account for potential nestedness, which in the site comparisons was really attractive. And so if we have the examples shown below with site D, E, and F, the Simpson beta diversity index would be 0 for those, meaning that it recognizes them as compositionally identical because the null hypothesis is that 2, E and F, are just subsets of the richer one, D. And so it's a really conservative measure. If you do see differences in beta diversity, even using such a conservative measure, we are pretty confident they actually have some biological reality to them. So with our beta diversity metric chosen, we can look for 2 types of patterns. One would be called biotic provincialization, where beta diversity is increasing through time, meaning these 3 regions, the Afar, Turkana, and the Southern Rift, are becoming more dissimilar through time. Their species pools are becoming more dissimilar through time. On the other hand, we have biotic homogenization, which is a lowering of beta diversity through time, where the species pools are becoming homogenized. They're sharing more and more taxa through time and may ultimately form a single regional species pool where all of the taxa are present in each of these three areas. When thinking about provincialization versus homogenization, we wouldn't expect all of the large mammal taxa to respond in the same way. In an environmental change that is advantageous for a grazer like a zebra is probably disadvantageous for the dispersal of a frugivore or a forest dweller like the monkey, or maybe a mixed feeder that is feeding on both grass and browse. So all of these animals on the screen are savanna mammals. They are representative of the types of animals we have in our data set. But because all of them, or each of them possesses a different environmental requirement as defined by their ecological niche, we don't expect them all to respond the same way in terms of showing higher degrees of provinciality or homogenization through time. And we'll come back to that. So if we are going to see biogeographic change in Eastern Africa over the last 6 million years, what would we expect some of the major drivers to be? Arguably, the most consequential change to African ecosystems and tropical ecosystems globally over the last several million years is the expansion of C4 dominated ecosystems. So in Eastern Africa, these are ecosystems where there's a significant amount of grass cover. We can track C4 expansion in a few different ways. So the plot I've shown here to the left is looking at molecular biomarkers from leaf waxes derived from drill cores all around the Horn of Africa. Those are the shapes labeled 228, 232, etc. You see at the bottom on the map. And using those isotope data derived from the leaf waxes, we can actually calculate the percentage of C4 biomass in Eastern Africa through time. And you can see, and I've pointed it out here with the arrow, that after about 10 million years ago, we have this major shift towards grassy ecosystems where we go from a landscape that's maybe 20% C4 grasses to one that's 60 or 70% C4 grasses. So that's a monumental change in the structure of Africa's terrestrial ecosystems. Another way to look at it is directly through terrestrial records. So a lot of these fossil sites, we have paleosols, ancient soils, that we can do geochemical analyses with to reconstruct the proportion of woody versus grassy vegetation through time. So if you do that in the East African Rift System, and then using some equations by Terry Sterling and colleagues, classify the proportions of ecosystems represented through time, so forest, woodland, open woodland, wooded grassland, grassland, we can see how fundamental the change to East African ecosystems was, in this case, over the last 6 million years. And so starting around 3 million years ago, you can see open woodlands, wooded grasslands, grasslands. These are all of the types of habitats that have substantial amounts of grassy cover. They really start to take off around 3 million years ago. And so in thinking about how animals with different functional traits, different diet, different body sizes, how they might respond to that expansion of C4 ecosystems, we made two simple predictions. To the left is where we start around 6 million years ago. Because C4 ecosystems are relatively less abundant, we expect there's lots of forest cover, meaning taxa dependent on forest resources like frugivores or arboreal taxa should be widely distributed among these three rift basin areas. And the grazers, because their preferred resource is not widespread, are the ones that show high endemism at this time. As C4 expansion, grassland expansion happens, we can move to the panel to the right, where we expect these patterns to flip. Now grazers, because of grassland expansion, are expanding their ranges. It's facilitating exchange of grazing taxa among these rift basin areas, and our browsers and other taxa tied to forested environments, the frugivores, they become more endemic through time. And so that was the simple set of predictions we had going into the study. And so what did we find? So this is the big picture pattern. To the left on the y-axis, you have beta diversity. One, remember, is maximum differentiation, more provincialization. Zero is minimum differentiation, where every community is redundant. You have maximum homogenization. What we see is that starting around 6 million years ago, Eastern African large mammal communities were pretty provincial. They were comprised of species not found in other regions, and that this pattern weakens through time, where we end up, by the Middle Pleistocene, middle to late Pleistocene, with faunal communities across the Afar, Turkana, and Southern Rift that actually look really similar to one another. We can ask, well, what was the role of different functional groups in driving that decline in beta diversity through time? And so each one of these panels shows you the last 6 million years. The gray points and the smoothed line through them show you the overall regional trend. And then the bars show you the trend for a particular functional group. So we can see if we look at the purple bars for browsers and frugivores, they mirror the regional trend through time. They start out with higher endemism that weakens through time. Grazers do the same thing, and some do it even more dramatically. So down at the bottom right, size 5 taxa, these giant mega herbivores, over 1,000 kilos, they show a really strong pattern of declining beta diversity through time. But what drove that decline in East African beta diversity? And there's actually two ways this can happen. Um, you can lower beta diversity through time by having more species shared between the regions. So if species are moving from subregion 1 to subregion 2, that is going to lower beta diversity through time. That could be what's happening. But at the same time, if nobody was moving between the regions, but we were just losing endemics within each community, that would have the same effect of lowering beta diversity through time. And so what we wanted to do was figure out a way to additively partition the effect of shared species gains versus loss of endemics through time in driving biogeographic change, that overall decline in beta diversity. And so this is some math that Andrew Dew came up with. If you're interested in learning more about it, you should definitely bother him with an email. But Andrew was able to partition out the roles of gains in shared species versus losses in endemic species through time and driving beta diversity change. And so what we have in these panels are different functional groups. So all the way to the left are carnivores. The top box shows us changes in shared species. The bottom box shows us changes in endemic species. And a few things pop out. We are losing endemic browsers and frugivores, endemic mixed feeders, endemic grazers. But the only group that's really gaining more shared species through time are grazers. And I've highlighted that uptick with these arrows here. We can then look at body size and again see some interesting changes. We lose size 5 endemics, these really large bodied 1,000-kilo mega herbivores. And the only taxa that gain in shared species are medium-sized things, size 2, 18 to 80 kilos, size 3, 80 to 350 kilos. So what's going on? How can we synthesize all that we've learned from those plots we just walked through into a sort of a coherent narrative of Eastern African biogeography during hominin evolution over the last 6 million years? So with the big picture, going back to 6 million years ago, we find that all of our faunal communities show a high degree of endemism, and that dispersals of large mammals and probably hominins between these regions was constrained by something at that time. We expected browsers and frugivores to be very widespread early in time as wooded environments are more dominant. We didn't find that to be the case. In fact, across the board, all of the functional groups show really high endemism up until about 3 million years ago. And that was a bit bizarre. You would think some sort of dispersal corridor that was environmentally favorable for one of these groups would have existed. But we didn't find that to be the case until we dug into the patterns a little bit more. And in fact, the only large mammal group that shows a different pattern, and in fact, the complete opposite pattern, are hippos. Early in time, hippos are really cosmopolitan. You find the same hippo species, for example, in Afar and Turkana up until about 3 million years ago. And then after 3 million years ago, each one of these subregions starts to host their own endemic hippo fauna. And so that suggests something really interesting is going on hydrologically. We can also look at this through the fish faunas, and we know that in the late Miocene and early Pliocene, a lot of the fish faunas are shared among these subregions. And so that suggests to us that there are really strong hydrological connections between these subregions early on time before large scale aridification of East Africa. And that hydrological network is what facilitated the dispersal of the same hippotaxa and the same fish taxa among these subregions, and at the same time constrain the movement of terrestrial large mammals between these subregions. We know, for example, that rivers pose very strong dispersal barriers to primates, including great apes. For example, bonobos being south of the Congo River, chimpanzees being to the north. And so if we imagine that between 6 and 3 million years ago, rivers are running down the main Ethiopian rift into the Turkana Basin and then through the rest of the Gregory Rift, that perhaps the existence of those hydrological corridors combined with the rift topography, that might have constrained species dispersal between these regions early in time for terrestrial taxa. And that could explain, or I guess I should say it doesn't explain, but it would not make it surprising if that was the case, that potentially the Afar and Turkana and the Chad Basin all hosted a distinct hominin taxon, because that's very much what we see the other large mammals doing at this time. On the other hand, after 3 million years ago, we see beta diversity really begin to decline. And that's basically driven by the loss of endemics across various functional groups and gains in shared grazers. So the animals that are actually moving between these subregions, becoming more and more shared through time, are the grazing taxa, and particularly medium-sized grazers, things like bovids and suids and horses, etc. That again is interesting because, to jump to a different hominin taxon, we have Homo erectus, which is so widely distributed in Africa. It's found throughout the Horn of Africa, down arguably into South Africa, and it's also in North Africa. And of course, we have out of Africa one, it's in Eurasia. And a lot of the time that we talk about Homo erectus and its massive geographic distribution, we link it something to its innate biology, that the paleobiology of this species must have been different in some way that made it able to disperse more widely than any hominin had done before. But if we look at that in the context of what's happening overall, at least in Eastern Africa, it's really not surprising. All the other mammals are doing the same thing, at least the grazers, at the same time. And maybe it has something less to do with the paleobiology of Homo erectus itself. And instead, hominins are just mirroring this broader change to East African fauna as we see at this time. Just an idea. Again, we went into this study not looking to provide answers to these key issues in hominin evolution, but just with the realization that having some sort of backdrop, some sort of context about what the other large animals are doing at this time may be a way to at least indirectly shed light on some of the patterns we see in the hominin fossil record. And so if you're interested in the study, it was published last year in Nature, Ecology, and Evolution. I didn't put the title up at the beginning of the talk because it basically gives away the take-home message of the talk. I'm happy to chat more about that. Thank you for the invitation. Thank you for all of the wonderful co-authors on this study, and I'm happy to answer any questions.
Thank you very much, John. That was really, really great. I'm sure we'll have lots of questions coming in. Would anyone like to ask a question, either in the chat, or you're welcome to put your hand up and ask out loud? Oh, Matt, you have a question. Go ahead.
Yeah, hi. Thanks very much, John. That was really interesting, and I'm going to go and seek out the paper. Um, I was just wondering, your graphs, from as far as I can tell, suggest this isn't the case. But I was just wondering if there's some relationship between the shared species and the endemic species, in that there might be competition between them. So, is it? Would you expect in some cases that when a species is able to move between these regions, it might displace an endemic species in the sense that it shares to a substantial degree the same niche as that species?
Yeah, absolutely. That certainly could be happening. I think I should have mentioned we analyzed the patterns in these 500,000 year bins. And so I think at the resolution we have, it's hard to actually document that replacement. Absolutely, if you get the extinction of the endemic, it vacates. Well, either you can have a disperser coming in and driving an endemic to extinction, or you can have an endemic in theory disappearing, and then that's why the disperser shows up in the region. Probably both of those processes are at play, but I don't think, at least with this more long-term record, we can get at that. If we had finer resolution data, like if we were to do this for the middle to late Pleistocene, you may be able to tease apart those patterns of replacement.
Okay, thanks.
Yeah, thank you.
Larry, do you have a question?
Yes, I do. John, thank you very much. I really enjoyed that. And I'm particularly intrigued by the hydrological barrier early on. But that's not the question I want to ask. But the question is more of an observation. You showed um, data from Serling's work.
Yep.
Yeah, and it showed about 500,000 years ago there was an increase in wooded grassland at the expense of grassland. And that I asked that because I'm draw that forward because I'm interested in this particular period. Um, could you talk us through a little bit what might be the implications of that shift from more dominance of grasslands to wooded grassland in terms of species diversity or homogenization, whichever direction it might be moving?
Yeah, my first response is I'm not sure. And then one other thing to keep in mind with this figure is that for each one of these stacked bar plots, most of the subregions are represented, like data, Paleosol data from each of the three subregions goes into a particular bar. When you get out to younger than 1 million years ago, the record becomes really sparse, where you have a little bit of data from the Afar and a handful from Oligocyle. And so before thinking about how that blip, that expansion of wooded grassland at the expense of grassland, what effect that might have had, I'd be interested to know what sites with Paleosol data are going into that, because I wonder how regionally representative it is, if you know what I'm saying. It may just be a
Okay. Yeah.
yeah. And that's something when we were writing the paper, I thought, well, do we write about this? Do we put it in the SI? We have the Paleosol compilation there. And so, like I said, we tried to get representation across the three subregions through time, but inevitably there's gappiness in the record, especially in the younger side here.
Okay. So that means you really can't say with any kind of certainty what might be happening at this time.
No, I would just be making it up, but I'm happy to go back and look at the data and dive more into it because if it's a real pattern, it's pretty interesting. It's a large reversal back to more wooded environments there.
Yeah. Yeah.
Yeah.
Okay. Thank you.
Thanks, Larry.
I'll just read a question that's come up in the chat from Gervais. She doesn't quite know how to word it, but if you did a follow-up study on just the hominins and if you had tool data, would you expect an increase in the similarity of tools across the three areas from less to more similar as species diversity diminished?
I don't know because I don't know. I don't know much about archaeology, despite being in a department of archaeology. Um, if it fits in with cultural traditions becoming more shared, there's more connectivity, then yes, I'd expect the archaeology to mirror the same patterns that we see in the fauna. Certainly the hominins mirror the same patterns we see in the large mammal fauna. So if that change in the distribution of the hominin taxes any reflection of how connected they may have become later in time, then I think the tools follow that, but I'm probably the wrong person to ask. It's a good question, though. Somebody should do it.
Thanks. That's a good answer. Um, I'll just wait and see if anyone else has any other questions. There are just a couple of people saying thank you in the chat for a great talk.
Thank you.
I have a question that it might not be quite in your area because it's kind of zooming forward through time a little bit. But
Okay.
people are starting to favor a structured meta-population model for the origin of Homo sapiens. So do you think looking at the faunal context of that would sort of shed any light on that argument?
Yeah, I do, because I think even later hominins, I mean, modern humans are pretty special, but in many ways, they're just responding to the same factors, same environmental drivers and patterns that the other large mammals are. And so as you get the push and pull with Green Sahara and then aridification and then the humid period comes back, we know that a lot of the sub-Saharan African mammals are moving up into North Africa and then contracting. Some get stuck up there, like blue wildebeests are known in North Africa for a few thousand years, and they're probably the hominins are probably just following the same routes that the other large mammals are. And that was sort of the whole rationale for just looking at the large mammals here is, especially in the earlier part of human evolution, functionally they should be responding the same way that a lot of the other large mammals are. And so, yeah, I think if you were interested in that population connectivity with modern human origins, looking at the large mammal faunas continentally across all of Africa would be really interesting. And it's a really data-rich period because you have all the archaeological assemblages versus here where the paleo assemblages aren't as rich. And so that's definitely a project worth doing, would be to model biogeographic connectivity in large mammals across Africa through time, say for the last 300,000 years, and see how that fit with the archaeological records or what we have of the modern human fossil record at that time.
Great, thank you. I'll put a pin in it for a postdoc if someone doesn't get to it before me. I'm not seeing any other questions coming up in the chat, so last chance if anyone wants to put their hand up. Oh, Matt, do you have another question?
Yeah, sorry, it's just a short thing related to the question in the chat. In terms of applying sort of thing to archaeology, do these diversity measures, are there sort of continuous equivalents of them, or do you need to partition things into discrete regions as you've done?
The the you mean discrete in terms of the like subregions or discrete in terms of the taxa themselves?
In terms of the region, so whether you could do this continuously across space.
Yes, you absolutely can. Your communities can have any sort of distribution. You don't have to aggregate them into subregions like we did. The reason we did it is because we wanted to be able to look at that 6 million year window. And so we restricted our sample to subregions where we knew we could hit that window continuously. Because if you have, you can imagine if you have changes in your sampling area through time, changes in beta diversity may just reflect you sampling a much larger region at period A and a much smaller one at period B. Yeah. But if you had sites across Africa that had a consistent um, spatial structure through time, it would be no problem to analyze it with these beta diversity metrics.
Right. Okay, thanks very much.
Yeah.
Great. I'm not seeing any more questions, so I think we can leave it there. So I'll just say thank you everyone for attending and attending all of our webinars so far this academic year. And thank you again to our speaker, Dr. Rowan, for a really great end to it all. So thanks, everyone. Just a couple more messages in the chat saying thank you.
Thank you.
Great.
Right.
Great. Thanks, everyone. Bye.
Take care.
Bye.